So I wanted to begin with this talk as speaking as a clinician who cares for people after cancer chemotherapy as they grapple with the long-term effects of our therapies. And before describing the syndrome, I want to say to begin with that cancer therapies have improved in recent decades tremendously. They are oftentimes life-saving and very important, but they don't come without a cost, and that cost is frequently a neurocognitive one. Frequently, after cancer chemotherapy, both in childhood and in adulthood, patients are left with fairly debilitating symptoms of memory dysfunction, both encoding new memories as well as difficulty with working memory, difficulties with attention, concentration, multitasking. The overall speed of information processing becomes hard. There's issues involving fine motor skill use and executive functions such as calculation. Because of this spectrum of neurocognitive dysfunction after cancer chemotherapy, often children require special education following their therapy, and adults find that they're not able to go back to their previous level of occupational function. So this is a big problem, and it's a big problem as we do better in treating these diseases effectively in terms of survival. It becomes an increasing issue for a great number of people. One example, which is a very important one, particularly because of the prevalence of the disease, is the cognitive dysfunction that women experience after breast cancer therapy. Studies have now shown that women as late as 20 years after chemotherapy for breast cancer continue to struggle with ongoing deficits in executive function, memory, and information speed processing. So this is a big problem. It's one that as clinicians and as scientists we need to understand so that we can address for our patients. I wanted to begin here with an outline. We'll start the talk by discussing the postnatal developmental processes and ongoing processes of neuroplasticity that can be affected by cancer therapy. And then we'll discuss what we know about the cellular etiology of cognitive dysfunction, particularly focusing on memory dysfunction that results from hippocampal stem cell dysfunction and ongoing loss of hippocampal neurogenesis in the childhood and adult brain, and the damage that cancer chemotherapy and radiation therapy confer to the myelin-forming precursor cells that are so important both in the protracted process of neurodevelopment that humans undergo as well as in ongoing mechanisms of neuroplasticity. So starting with a consideration of postnatal brain development and plasticity. In the young brain, and by young I mean the childhood and adolescent brain, there are numerous processes that are ongoing and often modulated by experience that contribute to formation of an adult human nervous system. There's the formation and stabilization of synapses or the connections between nerve cells. There's subsequent pruning back of exuberant connections that are made. There's ongoing postnatal neurogenesis or the production of newborn neurons, especially in the hippocampus, which is a structure critical to normal memory function. And importantly, there's ongoing gliogenesis, or the production of the glial support cells in the nervous system. And we'll spend a fair bit of time talking about that. Those glial cells contribute to the maturation, function, and plasticity of the white matter infrastructure that's really critical for normal circuit function in the brain. In adulthood, there continues to be plasticity of synapses or connections between nerve cells. There's ongoing generation of newborn neurons in the hippocampus, and there's plasticity of that white matter infrastructure with ongoing generation of new oligodendrocyte glial cells. Many of these processes depend upon the normal function of postnatal stem cell populations in the childhood and adult brain. And so just to highlight a few of those, critical true stem cell populations, and by stem cell I mean a cell that can self-renew and generate all of the progeny of that organ in the brain that includes neurons and both types of glial cells, astrocytes and oligodendrocytes. In the subventricular zone, there is a stem cell population which contributes newborn neurons to the olfactory bulb and populates the white matter infrastructure I had been mentioning with these myelin-forming precursor cells, or oligodendrocyte precursor cells. In the hippocampus, there's also a stem cell population which continues to generate newborn neurons and, to a lesser extent, ongoing generation of newborn glial cells. On the left-hand side of this diagram that I'm showing you, there are cognitive processes which, in a simplified way, are kind of localized to the regions that you see here. And so episodic memory function depends very importantly upon the hippocampus and upon the normal function of these hippocampal stem cells, while working memory, executive functions like calculation, attention, concentration, speed of information processing, our ability to multitask all depend upon cerebral structures, heavily on the frontal lobe structures, and really depend critically upon the integrity of this white matter infrastructure connecting important cortical regions. Speaking a little bit more about hippocampal neurogenesis, which derive from those hippocampal stem cells, hippocampal neurogenesis is important for the encoding of new memories and for many types of hippocampal function. We know that neurogenesis in the hippocampus increases with healthy things like physical exercise, with being in an enriched environment with cognitive challenges. And we also know that increasing hippocampal neurogenesis correlates positively with performance on hippocampal-dependent behavioral tasks and experimental systems. Conversely, decreasing neurogenesis, and there are many things, unfortunately, that do that, many of which are part of cancer chemotherapy and treatment, decreasing neurogenesis with glucocorticoid or steroid exposures, chemotherapeutic exposures, and irradiation that involves the hippocampus impairs memory, it impairs function on hippocampal-dependent memory tasks. Myelination is a really fascinating process which extends over 3 decades of postnatal life. So people don't finish developmental myelination actually until at least their mid-30s, and there's now evidence that there's ongoing myelin remodeling throughout adult life. Considering the pattern of myelination, which is so important for the normal function of nerve cells, and you see a myelinating oligodendrocyte here on the left side of the screen extending its plasma membrane to wrap and thereby insulate surrounding neuron projections, which are called axons. This is critical for normal circuit function in the brain. And the pattern of myelination in development follows a very reliable and predictable topographic and chronological order. Proximal structures myelinate before distal ones, the sort of center of the brain myelinates before the poles, the cervical spinal cord myelinates prior to the lumbosacral spinal cord, and for those of you who have kids, this is something that you can almost observe over the first year of life. Babies can first pick up their heads, then they can move their hands, around 6 months they can sit up, and then finally around a year they can walk, and that progression is due largely to this proximal to distal pattern of myelination down their spinal cords in the first year of life. Proximal structures, as I said, myelinate prior to distal ones, sensory circuits prior to motor circuits, projection neurons prior to association neurons, and in general, more basic functions like movement kind of come online with myelination before more complex, higher cognitive functions. And interestingly, the neural projections that are most critical to higher cognitive function, such as association pathways between cortical regions, these areas myelinate last and remain incompletely myelinated, it's almost suggesting that there could be some tunability of the degree to which they're myelinated. And that kind of brings up this idea that myelin might in fact be plastic, that there might be some remodeling of myelin that is experience-dependent and functionally relevant. And there's some evidence for that in the adult human and pediatric human neuroimaging literature. So if you teach an adult, for example, to juggle, you will see a change detected by diffusion tensor imaging, this non-invasive MRI modality, in correlates of myelination within hand-eye region relevant white matter, which is really intriguing, suggesting that the experience of learning to juggle actually changes the structure of the myelin within relevant active circuits. The idea that myelination in the childhood and adult brain might be a plastic process, might be adaptive, and might be a functionally relevant dimension upon which nervous system plasticity occurs is something that has been discussed in the literature for several decades, but had not been really conclusively shown. And so my laboratory, in work funded by CIRM, used in vivo optogenetic strategies in rodent models to demonstrate that behaviorally relevant neuronal activity, we chose to utilize the premotor motor planning circuit, results in robust proliferation of those myelin-forming neural precursor cells called oligodendroglial precursor cells, subsequent differentiation of those cells, and increases in the degree to which the active circuit is myelinated. Further, we found that this resulted in a lasting change in the function of this activated circuit, such that motor function was actually improved in a way that depended upon the formation of these new oligodendroglial precursor cell-derived myelin-forming cells. Subsequently, a group in England, Bill Richardson's group, has shown with a very elegant experiment that if you block oligodendrocyte precursor cell differentiation, specifically in adulthood, animals are unable to learn new motor tasks. And so there does seem to be this role for myelin-forming precursor cells in learning and in neural function and neuroplasticity in general. So in many ways, we see that neural precursor cells are thus central to normal cognition and normal neurological function. We know that neural stem cells, particularly in the hippocampus, need to be intact and able to generate newborn neurons in the hippocampus. We know that the function of these oligodendroglial precursor cells within the white matter infrastructure are critical for myelin plasticity in the developing and also in the adult brain. And so given that there are so many precursor cell populations within the developing and adult brain that are important for normal cognition, it stands to reason that cancer therapies which target dividing cells, which target to which precursor cells are fairly sensitive, might be an important etiology for the long-term cognitive effects of cancer therapy. So I want to tell you a little bit about what happens in the hippocampus following cancer therapy. Following cranial radiation, chemotherapy, people can exhibit devastating neurocognitive symptoms, yet on standard neuroimaging scans look fairly normal. So that indicates that there's some subtle process which is susceptible and responsible for the clinical deficits that we see. When we look with more sensitive and functional neuroimaging technologies, we see that, for example, following childhood leukemia therapy, there is both structural and functional disturbances in the hippocampus in adulthood. Examining post-mortem tissue samples, we've shown that hippocampal neurogenesis is dramatically impaired in children who've received cranial radiation therapy and chemotherapy for childhood brain cancer. And so in contrast here on this slide to children who passed away from non-neurological and non-oncological causes, you can see that there is a baseline level of hippocampal neurogenesis is detected in this postmortem tissue by the neuroblast marker double cortin. There's a high level in infancy, which then tapers off to a baseline level that continues throughout life. In contrast, these children who had undergone treatment for medulloblastoma, as well as an adult who had undergone treatment also for medulloblastoma, you see this absolute bottoming out of detectable hippocampal neurogenesis in this tissue, confirming what we've examined pretty closely in animal model systems, that hippocampal neurogenesis appears ablated following chemotherapy and radiation therapy exposure. Work that was done during my graduate years with fellow CIRM grantee Theo Palmer at Stanford, we had found that cranial radiation therapy impairs hippocampal neurogenesis, interestingly, by damaging the microenvironment that normally supports precursor cell function. The stem cells actually are there. They're quiescent cells. They tend to repopulate in the hippocampus following radiation exposure, but they are unable to differentiate into newborn neurons due to a radiation-induced microglial inflammatory response that persists in that stem cell niche. In addition to the effects of radiation therapy on hippocampal neurogenesis, chemotherapy further targets hippocampal stem cells and alters both stem cell integrity and the neurogenic niche within the hippocampus. Focusing then on the cellular etiology of myelin-forming precursor cells and disrupted myelination following cancer therapy. There's a great deal of evidence in the neuroimaging literature, again, that structural deficits in white matter exist following chemotherapy exposure in both childhood and in adulthood. And we've just discussed the role of postnatal gliogenesis from these glial precursor cells. We know that Olgodendrocyte precursor cells contribute to ongoing myelination, which is important for normal cognitive maturation, maintenance, and ongoing plasticity of functional neural circuits. And so it stands to reason that disrupting the function of those precursor cells would result in a range of neurocognitive deficits, and actually we would predict exactly what we see following chemotherapy exposure and radiation therapy exposure that affects the cranium, including poor attention and concentration, slower information processing, poor memory retrieval, and poor multitasking. Animal models have shown very nicely that chemotherapy and radiation does deplete oligodendrocyte precursor cell populations. And going again back to human tissue, we find in examining post-mortem tissue from children who had undergone chemotherapy for non-central nervous system malignancies that there is a decrement in the detectable oligodendrocyte precursor cell populations. What we found is that this was in a very striking and unexpected pattern. We saw a very important decrease in the concentration of these precursor cells in subcortical white matter, but it was restricted to subcortical white matter. The gray matter oligodendrocyte precursor cell population was intact, suggesting an important aspect of heterogeneity between these populations. Using mouse models to recapitulate this in a murine model of juvenile chemotherapy exposure, we find the same thing, that there is this depletion of subcortical precursor cells with preservation of the gray matter oligodendrocyte precursor cells. And so taken together, cancer therapy-induced depletion of oligodendrocyte precursor cells, both in childhood and adolescence, as well as later in adulthood, would prevent normal developmental myelination of brain regions important in cognition. It would also, we would predict, prevent the adaptive changes that occur in myelin that appear to be critical to normal cognitive function and learning. And so what are our therapeutic options, understanding the cellular basis for some of the cognitive deficits that we observe in our patients? Certainly in the clinic, we can use symptomatic therapy. Educational interventions and memory aids are helpful for patients who are struggling with neurocognitive deficits after cancer therapy. Stimulant therapy like methylphenidate can really improve speed of information processing and can offer patients symptomatic relief. A lot of the patients that I care for really depend on methylphenidate to get through their school day and to get through their work day. To address the memory dysfunction, one of the drugs that is used frequently in Alzheimer's dementia, acetylcholinesterase inhibitors, has proven to be useful for memory dysfunction following cancer chemotherapy and radiation exposure. But what we really want to do is to fix the problem. We want to regenerate these populations that are important to maintain the structural integrity of the brain and to promote plasticity. And so strategies aimed at promoting hippocampal neurogenesis are actually currently in clinical trial based on our previous findings that microglial inflammation impairs hippocampal stem cell differentiation and ongoing neurogenesis. There are ongoing clinical trials of anti-inflammatory therapy during and after radiation. And aerobic exercise, which we know to increase hippocampal neurogenesis. There's actually a really lovely trial at University of Toronto using aerobic exercise to promote hippocampal neurogenesis after childhood brain tumor therapy, which is showing some real promise. And regenerative therapy aimed at replacing the oligodendrocyte precursor cell populations or promoting their ability to remodel that white matter is an important challenge. And in CIRM-funded work, my lab is focusing on trying to harness the molecular mechanisms that govern this activity-dependent myelin remodeling, which we've observed in the healthy brain to promote white matter regeneration following cancer chemotherapy exposure. To summarize, cancer therapy, while critically important for preserving life, does frequently cause long-term deficits in cognitive function, importantly in memory, attention concentration, and executive function. Damage to susceptible neural precursor cell populations is an important etiology underlying these deficits. Symptomatic management can be offered right now for patients who are struggling with these long-term problems, and we hope to develop effective regenerative strategies moving forward. I want to thank CIRM for supporting so much of this work in my laboratory, and all of the people in my laboratory who have been working hard on these projects.